Welcome back to this next video, and in this video we are going to talk about the protein microarray, which is actually a high throughput screening method for the study of gene expression. So the first thing is, what is a microarray? Now if you look at this term microarray, it is actually made up of two terms. One that is the micro and the other one that is array. So the first thing I want to focus on is array. Now what are arrays? Arrays, they are solid support upon which a collection of gene-specific probes, they have been placed at defined location either by spotting or direct synthesis. For example, if this is the solid support and 9 spots that are available on this particular solid support, so if you can see over here, this particular spot is referring to the presence of the gene number 1 or the protein number 1. This particular spot is referring to the gene number 2 or the protein number 2. This particular spot is referring to the gene number 6 or the protein number 6, and this particular spot is referring to the gene number 9 or the protein number 9. What this means is that at each and every location of this particular spot, you know that what is present, and if there is detection on a particular spot, you can actually identify the presence or absence of a particular if there is detection, that means that that particular protein or that particular gene is present in your sample, or that is expressed in your sample. Now the second component of this microarray, that is micro. Now this term micro, it actually refers to the small size of the solid support. So if you look at these chips, whether that is the DNA chip or that is a protein chip, they are actually small, therefore they are called as the microarray. So this term microarray means that an array has been synthesized on a small solid support. Now the arrays, they actually come in two flavors. One that is known as the DNA microarray, which is actually an indirect method for the study of the proteomics, and the other one is the protein microarray, which is actually a direct method for the study of the proteomics. Before going into the protein microarray, I want to focus on two things. That when you talk about the study of the proteomics, the methods that are used, they are broadly divided into two classes. One type of the methods, they are known as the indirect methods, and the other type of the methods, they are known as the direct methods. Now in the indirect methods, we study the presence or absence of a particular protein, focusing on the precursors of those particular proteins. And you all know that the precursors for the proteins, they are the messenger RNA. So in indirect methods, you are actually focusing on the presence or absence of a particular messenger RNA, and that particular messenger RNA is then showing you the absence or presence of that particular protein. And the most famous indirect methods that are used in the study of the proteomics, that includes the northern blotting and the DNA microarray. The other type of the methods that are used for in the study of the proteomics, they are the direct methods. And in the direct methods, you are not focusing on the precursors, you are directly focusing on the proteins. Therefore, these kind of the methods, they are known as the direct methods. And one of the most common method that comes under the heading of the direct methods for the study of the proteomics, that is the protein microarray. Now a protein microarray, sometimes also known as a protein chip, that is actually a high throughput method used to track the interaction and activities of proteins. So as you are directly focusing on the protein, so the protein microarray is a direct method for the study of the proteomics. So it is a high throughput method used to track the interaction and activities of proteins, to determine their function and determining functions on a large scale. And one of the main advantages of the protein microarray that lies in the fact that large number of proteins that can be tracked in parallel. What this means is that in a single experiment, you can actually focus on a large number of proteins, thereby making the protein microarray a high throughput method. If you talk about the solid support, or if you talk about the protein chip, a lot of things, a variety of the materials that can be immobilized on the protein chip based on the specific requirements. So the probes on the chip that actually come in a different variety, and a variety of things can be immobilized on the protein chip. Now, the most important probes that you can immobilize on the protein chip that include the antibodies, the antigens. So you can immobilize the antibodies. In other cases, you can also immobilize the antigens. You can also immobilize the aptamers, and these aptamers are actually the nucleic acid based ligands. You can also immobilize the Fe bodies on the protein chip. These Fe bodies, they are small robust protein engineered to bind to a large number of the these Fe bodies are small robust proteins which are engineered to bind to a large number of target proteins or peptides with high affinity, and they actually imitate the monoclonal antibodies and are therefore a member of the family of antibody mimetics. You can also immobilize the full-length proteins or their specific domains. So these are these things, these 5 probes, they can be immobilized on a protein chip, and that is totally based on the specific requirements. Now, if you talk about the sources of probes on chips, like if you can immobilize these 5 kind of the things, from where you can actually get these things. So the first source of the probes on the chip, they can directly come from the cell lysate. For example, if you are interested in the full-length proteins, what you can do is you can use the cell lysate, and by using different chromatographic techniques like the affinity chromatography or the ion exchange chromatography, you can isolate your desired protein from the cell lysate. And I will have a detailed video on the affinity chromatography and the ion exchange chromatography. But at this particular time, just keep in mind that one of the sources of the proteins that you can utilize as probes on the chip, they can come from the cell lysate, utilizing different chromatographic techniques like the affinity or the ion exchange chromatography. The second important source, they are the recombinant DNA technology. Now what you do is that this is actually a flow diagram what is happening in the recombinant DNA technology. For example, if you are interested in a protein which is actually a growth hormone, what you do is that you are going to isolate that particular gene from the human cells, and you are going to cut that with the restriction enzymes. On the other hand, what you do is that you are going to take the plasmids from the bacterial cell, and you are going to cut it with the restriction enzymes. And then what you do is that you are actually going to insert this gene of interest into this plasmid, and when you do this, you can actually insert your gene of interest into this particular plasmid. Then you are going to introduce this plasmid into a bacterial cell, and the bacterial cell is going to express that particular gene, giving you the growth hormone or a particular protein. So generally you can say that you can take your gene of interest, you can insert that into the plasmid, and you can actually insert that plasmid into an expression system which can give you the expression of that particular protein. If you are dealing with proteins that do not go for the post-translational modification, you can actually utilize the E. coli cells. But if you are dealing with proteins which go through the post-translational modification to get a particular 3D structure, you cannot use a prokaryotic cell like an E. coli. In these particular instances, you have to use eukaryotic systems like you can use the yeast which can actually provide you with the facility of the post-translational modification. So the second source of probes on the chip, they can be from the recombinant DNA technology. The third type of the source that is available, that is through the formation of the synthetic peptides. So you can actually make these peptides synthetically, and the most established method for the production of synthetic peptides in the lab is known as the solid phase peptide synthesis. So you can actually make these small peptides in the lab utilizing this solid phase peptide synthesis. Now what you can actually do is that when you have made these small peptides, you can actually link these peptides to each other to give you a larger peptide. Like if you can see over here, this peptide is like about 50 amino acids. This is about 50 amino acids. This is about 50 amino acids. But when you combine them, you can actually get a polypeptide which can give you the desired 3D structure, which you can use in the chip, which you can use on the chip for the detection of a particular protein. So this is the third type of the source that is available for the synthesis or for the synthesis of the peptides. The most important types that is available these days for the as a source of the probes for the protein microarray, that is the cell-free translation system. Now the cell-free translation system, also known as the cell-free protein synthesis, for short it can be known as the CFPS, is also known as the in vitro protein synthesis. That is actually the production of the protein using biological machinery in the cell-free system, that is without the use of the living cell. Still, you are making the proteins utilizing the biological machinery, but you are not using the intact living cells. Now the common components of a cell-free reaction that include cell extract. Of course you need a cell extract, you need an energy source, and the energy source that are commonly used in the cell-free translation system, that are the phosphoenol pyruvate, the acetyl phosphate, or the creatine phosphate. You need a supply of the amino acids because these will be used for the synthesis of your desired protein. You need cofactors like the magnesium, and these magnesium, they are very important for keeping the ribosome intact. You need the DNA with the desired gene, and the necessary cell machinery that include the ribosome, the amino acid transfer RNA synthetases, the translation initiation, and the elongation factor, etc. So what you do is that in the cell-free translation system, you are actually going for the production of proteins using biological machinery without the use of the intact living cells. But of course, you are using the machinery of the living cells. Now the cell-free translation system that come in a variety of the methods. Now one of the most common method that is used in the cell-free translation system is known as the nucleic acid programmable protein array, which is for short known as the NAPPA, or you can call it as NAPPA. Now what you do is in the nucleic acid programmable protein array, that you have got a slide surface, and on this slide surface you are actually immobilizing an avidine protein. You are actually attaching the avidine protein to the solid surface, and your template DNA, you are actually adding a biotin molecule to your template DNA. So you are doing two things. On the slide surface, first you are adding the avidine, and you are adding a biotin molecule to your DNA template. Now then the biotin and the avidine, they are going to interact with each other, so you are indirectly immobilizing your DNA template sorry, on this slide surface. If you talk about the interaction of the avidine and the biotin, so this avidine is actually a tetrameric biotin binding protein, and if you can see over here, one avidine molecule can actually bind to 4 biotin molecules, and biotin is actually a water soluble vitamin. So your aim is to immobilize your template DNA on this solid surface, but you cannot do that directly, so you are indirectly immobilizing this template DNA on this slide surface. So you are attaching the avidine to the slide surface, biotin to your template DNA, and when the biotin and avidine interacts with each other, the template DNA that have been immobilized on the slide surface. Now one of the important things to keep in mind is that when you talk about this template DNA, it is not purely coding for your protein of desire. It is actually a hybrid DNA which contain a part for the GST protein as well. We will come to that in a while, that why we are adding a part of the GST DNA to our template DNA. But this template DNA is coding for your desired protein plus part of the GST protein. The other important thing that you are immobilizing on your solid surface besides the avidine is the anti-GST antibody. And this anti-GST antibody actually have the ability to bind to the GST. So you are actually using the GST part in the template DNA so that it can interact with this antibody, because this is anti-GST antibody, and by doing that you are actually immobilizing your target protein on this slide surface. So once you have immobilized your template DNA, once you have attached the anti-GST antibody, the next thing you do is that you are going to add the cell extract. When you add the cell extract is containing the transcription and the translation machinery, so that is going to transcribe their template DNA, and when it transcribed it, the ribosome that is actually going to translate that particular messenger RNA into the polypeptide chain. Now, if you look at this polypeptide chain, as I've told you, it is actually this DNA template is coding for two things. One is your desired protein and the other one is part of the GST. So if you look at over here, this red spot over here is actually the GST part, which is coded by the part of this template DNA, and the other part of this particular DNA is coding for your desired protein. Now your desired newly synthesized protein that have been immobilized to the solid surface via this GST tag through the anti-GST antibody. So once you have immobilized your newly synthesized protein, you can actually use this for the detection of the target in your sample. So this is one method which is known as the nucleic acid programmable protein array. Now this nucleic acid programmable protein array have got some drawbacks, and the main drawback of this method is the extra interiors preparation step at the beginning of the process. What I mean by that is that the first thing you have to do is that you have to clone the cloning of the cDNA in an expression ready vector. There is the need to biotinylate the DNA, but not to interfere with the transcription. Moreover, the resulting protein array is not pure because the proteins are co-localized with their DNA templates and the capture antibodies. So if you can see over here on this particular slide surface, now this antibody, that is not our thing of desire. We do not want this antibody, but we are using it to immobilize things. So this is actually a sort of the impurity in this particular slide surface. Another thing is that once the DNA that have been translated into the desired protein, we also do not require this DNA, but as that has been immobilized now by the Abelian biotin chemistry, therefore it is present in the well also. So this is also acting as a source of the contamination. So these are some of the drawbacks of the nucleic acid programmable protein array. Now another important method of the cell-free translation system is known as the protein in situ array. And this protein in situ array, it is actually a modification of the NAPPA. And unlike the NAPPA, PISA completely bypasses the DNA immobilization, as the DNA template is added as a free molecule in the reaction mixture. So as you can see over here, you have got this slide surface, you've got this protein tag capturing agent. It can be the antibodies. You can also use the other capture agent as well. So what you do is, to this particular slide surface and hence this well, you add the template DNA and the cell extract. The machinery is going to transcribe this particular DNA template into the messenger RNA, and the messenger RNA is then going to make the polypeptide chain, and hence the newly synthesized protein that have been immobilized to these capture agents utilizing the tags over here. So this can be the GST tag, as we discussed in the nucleic acid programmable protein array. So if you compare this particular structure with the one that we see in the NAPPA, is that there is no immobilized DNA template present in this particular well or at this particular position of the slide. So this is the protein in situ array, which completely bypasses the DNA immobilization. So in the end, the DNA that will not be present in the well of the chip as a contamination. Now, when you talk about the protein immobilization on the solid support, once you have made your desired protein by utilizing any of the strategies that we just discussed, that you take that from the cell lysate, you use the recombinant DNA technology, you are going for the synthetic peptide synthesis, or you are going for the cell-free translation system. The next important step is the protein immobilization, because if you cannot mobilize them on the solid support, that means you cannot actually detect your target in the sample. So following the protein production, the protein immobilization on the solid support, that is also crucial. And this solid support, you can use a variety of them. You can use the glass slides, you can use the micro-titer plates. A variety of the protein immobilization solid supports are available. Now, the most common supporting material in use, that includes the aldehyde, the carboxylic esters, the nitrocellulose membrane, the polystyrene, the agarose, polyacrylamide gel, or the hydrogel. What I mean by that is that you will be having solid support, and on the solid support you will be having these supporting materials which will be used for the immobilization of the protein. Now, an ideal surface for the protein microarray fabrication or immobilization has to be capable of immobilizing the protein. This is the first important property, that a solid support should have the ability to immobilize the protein. But at the same time, if it is going for immobilization of the protein, it should also preserve the three-dimensional conformation of the protein, because after immobilization, if the 3D conformation of the protein that is changing, that means it will not be able to interact with its target protein, because the proteins, they actually have their specificity because of the specific three-dimensional structure. So if you are not preserving the 3D structure of the protein by immobilizing them on solid support, that means that this immobilization will be of no use in the experiments. So the first thing an ideal surface should do is immobilize the protein, and that it should also preserve the three-dimensional conformation of the protein. The solid support should also not change the chemical nature of the protein, because if the chemical nature of the protein is changed, again it will not be going to interact with its target in the sample, and you can actually get false positive or false negative results. Now, one of the important thing about the protein chip, and which is making it different from the DNA chip, that there are microvels present on the protein chip. Now, these microvels, they are very important because an aqueous environment is essential at all stages of the protein array manufacture and operation to prevent protein denaturation. Now, the protein, they are very sensitive to their environment, therefore you have to provide an aqueous buffered environment to the protein at all stages of the manufacture and operation to prevent protein denaturation, because if the proteins, they are denatured, they will be of no use in giving you any useful results. Therefore, sample buffers, they are used to provide optimum environment to proteins, and the humidity of the manufacturing environment is carefully regulated. Now, what these microvels do is that if there are microvels, if this is one of the microvel of a protein chip, so this microvel is having a dual advantage. One, it is providing a buffered aqueous environment to the protein. Secondly, it is also preventing the cross-contamination between samples. So if this is the microvel, this can be your protein of interest. So you're actually providing a buffered aqueous environment to the protein so that it do not lose its particular 3D structure. If you talk about the detection system, how are you actually going to detect your desired or your target in the sample? Now, in this particular image, I'm showing you a comparative thing, that how you can actually compare two samples by using a detection system for a protein microarray. So, for example, if this is your control and this is your sample. Now, what you do is that you are going to extract the proteins from your control, and you are also going to extract the proteins from your sample. The next thing you have to do is that you are going to label them. This is the labeling system that should be different for the two samples. So say for example, if you are having the proteins from the control, you are going to label them with a green fluorescent molecule. And once you have isolated the proteins from your sample, you are going to label them with the red fluorescent molecule. When you have labeled them, when you have labeled both of the proteins from the control and the proteins from the sample, you are going to combine them and you are going to add them onto the protein chip. When you add them, these proteins are going to bind to their specific antibodies. In this particular case, they are showing you that the antibodies, they have been immobilized on the solid support or on the protein chip. So once you add both of these samples are going to interact with the proteins that are present on the on this particular protein slide, and they are going to bind specifically. What I mean by that is, say for example, if this is the antibody number 1 and this antibody number 1 is only interacting with these blue cones with a red fluorescent molecule, that means that these antibodies are going to show you the detection that the proteins are expressed only in the sample and they are not expressed in the control. If you talk about this one, this particular image is showing you that these particular proteins which have been labeled with a green one, they have been collected from the control sample. So here only the yellow proteins, they are interacting. Now if you look at over here, here at this particular position only the red fluorescent is present. What you mean by that is that if you go through the laser system that I'll discuss in detail in the DNA microarray, so what you do is that when you have added your samples to this protein chip, you are going to apply different lasers, and those particular lasers are then going to tell you about the presence of the particular protein at particular position. Just for example, if you see over here the red fluorescent molecule that is present, so if you are applying a red laser over here, that is going to give you the fluorescence, and you will see that this particular protein which is interacting with this particular antibody is only expressed in the sample condition because there is only a red fluorescence. If you talk about this green one, this means that only these kind of the proteins, they are only expressed in the controlled samples, because the controlled samples, they have been labeled with the green fluorescent molecule. Now if you are getting a yellow one, what this means is, if you look at over here, some of the antibodies, they are binded with the green fluorescent molecule, some of the antibodies, they are binded by the red fluorescent molecule. That means that these proteins, they are expressed in both conditions, in the controlled condition and in the sample condition. So when you apply the red laser, you are going to get the red fluorescence. When you apply the green laser, you are going to get the green fluorescence, and when you combine the image, you are actually going to get a yellow one over here. So the yellow fluorescence will be showing you that this type of the proteins, they are expressed in both conditions. So when you talk about the detection system in the protein microarray, you are actually using the laser technology that had been used in the detection of the DNA microarray, and I will have a detailed video on the DNA microarray in the near future, where I'll show you the animation how these laser systems that actually work. So if you like the video, please subscribe to my channel, and please hit the like button and share it with your friends, and we will continue the discussion in the next video.